Hi, I'm Amy Botu.
I'm Vishnu Morella.
I'm Nikkil.
And I'm Mahaniya Srinivasan.
And this is the characterization of phages infecting Mycobacterium smegmatis. So why do we care about bacteriophages? It's important to study bacteriophages as they are a kind of virus that are able to infect and kill potentially harmful bacteria without any adverse effects on human cells. This ability makes phages a unique solution to certain bacterial infections, which may not have a clear antibiotic solution. As the effectiveness of antibiotics decrease, the phage treatment is proven to be a lot more helpful in those cases. About Mycobacterium. Mycobacterium is a genus of actinobacteria, and there are over 190 species in this genus. While our species of Mycobacterium, smegmatis, was not infectious to humans, there still exists many pathogenic species of this genus which can lead to illnesses such as tuberculosis and leprosy.
The methods that were used. We took an extract from a soil sample and mixed it with bacteria to see if there was a phage present in the soil sample. After a phage was found, we purified the phage and isolated plaques. These plaques are formed by bacteriophages injecting their DNA into Mycobacterium, which replicates the phages to create phage proteins, which are then assembled into phage particles, which then cause host lysis. These dead bacteria cause clear plaques in bacterial growth. We use several methods to isolate and characterize these phage samples, such as filtration and spot testing, serial dilutions, restriction enzyme digestions, electrophoresis of genomic DNA, and staining phage particles for transmission electron microscopy.
Using undiluted phage samples, we created plaque assays to obtain well-isolated plaques. This helps us to create a pure phage population as well as determining phage morphology. We then performed a high titer lysate that allows there to be sufficient DNA in the sample. Wet lab results were found using a transmission electron microscopy machine. Imaging was used to find a phage in the soil sample that was obtained by the students. The TEM imaging displayed a non-contractile tail, and the restriction enzyme digest showcased a double-stranded DNA sequence.
So after conducting the methods of filtration, spot testing, DNA and phage purification, and agarose gel electrophoresis, the final result was a DNA sequence. The sequence from the lab was then later sent to Pittsburgh to further analyze and obtain the complete genomic sequence as pictured on the slide. The sequence from the slide is now being analyzed and annotated for function and position.
We would like to thank all of you for your support. Specific thank yous to HHMI Phages program, the VCU Life Sciences program, and the VCU Nanomaterials Core Characterization facilities. We would like to give a special shout out to Stuti Shah and Harnoor Sandhu for their method images. Thank you.